Good day everyone, I am Desert and Francisco, and together with me are Mr. Rivera and Mr. Olimpayan, and today we will be presenting our post-laboratory discussion regarding on extraction of tannins from mint tea leaves. For the procedures and materials, we started by weighing 50 grams of dried mint tea leaves and then macerated them with 207 ml of distilled water, just enough to cover the sample. The mixture was left to stand for 24 hours to allow proper extraction. After that, the extract was heated in a hot water bath until it was completely dried, and the dried extract was used for further tests. Shown here are the photos documenting our process. We first weighed the mint tea leaves, then performed the 24-hour maceration in distilled water. After maceration, we filtered the mixture to separate the leaves and collected the filtrate. Finally, we heated the extract in a hot water bath to dryness, preparing it for tannin testing. Here are the data we obtained from the extraction yield. The weight of mint tea leaves used was 50 grams and the weight of the dried crystal obtained was 26.8 grams. Using the formula percentage yield equals to the weight of crystals divided by the weight of tea leaves times 100, we got a yield of 53.6%, and this indicates that more than half of the plant material contributed to the extractable components during our process. In terms of organoleptic properties, the extract showed a deep brown color, a thick syrupy semi-dry texture, and a semi-solid physical state, and these characteristics are typical for tannin-rich extracts, supporting the expected outcome of the procedure. For the solubility test, the extract was soluble in both water and alcohol but insoluble in ether, and this confirms the polar nature of tannins as they dissolve well in polar solvents but not in non-polar ones. For our first set of chemical tests, the ferrichloride test produced a greenish-black color indicating the presence of phenolic tannins. The 10% gelatin test is theoretical only, and although we didn't perform it in the lab, literature states that a white flocculant precipitate would confirm tannins. Lastly, the bromine test gave a dark gray color showing the presence of hydrolyzable tannins. For the second set of chemical tests, the lime water showed no significant color change, suggesting the presence of catechin tannins. Meanwhile, iodine test produced a dark brown color confirming the absence of starch in the extract. These results together reinforce that our extract mainly contained tannin compounds rather than carbohydrate impurities.
So moving on to the answers to questions part. So number 1, what are tannins? So tannins are natural polyphenolic compounds found in plants like bark, fruits, tea leaves, and nuts. So they have an astringent taste and can bind and precipitate proteins. So classified as hydrolyzable or condensed tannins based on their structure and reaction. So they serve as plant defense compounds and have important pharmaceutical and industrial use. Number 2, give the uses of tannin in pharmacy. So in pharmacy, tannins are valued for their astringent, antioxidant, and antimicrobial properties. So they aid in treating burns, sore throat, wounds, diarrhea, and minor skin irritations by tightening tissues and reducing inflammation. So tannins also protect damaged tissues with a protective coating and are used in antidotes for alkaloid poisoning by forming insoluble complexes with harmful substances. So number 3, to which type of tannin does gallic acid belong? Give its use. So gallic acid is classified under hydrolyzable tannins, which can be broken down by acid or enzyme into sugars and phenolic acids. So it is known for its antioxidants, antibacterial, and anti-inflammatory properties. So in pharmacy, gallic acid is used in the manufactures of inks, dyes, and medicines that help protect cells from oxidative damage. So it is also used in formulation for skincare and wound healing, making it valuable compound in both pharmaceutical and cosmetic products. Number 4, what is the use of tannin in plants? So in plants, tannins serve mainly as a protective compound. So their bitter taste discourage animals and insects from eating the plant, while their ability to inhibit bacterial and fungal growth helps prevent infections. So when a plant is wounded, the tannins form a protective layer that seals the damaged tissues and prevents further decay. So they also help plants survive in harsh environments by reducing water loss and strengthening plants' tissues. Number 5, give different pharmaceutical uses of tannin. So tannins have numerous pharmaceutical uses because of their healing, antiseptic, and astringent properties. So they are also in ointments, creams, and mouthwashes to treat wounds, burns, and sore and sore gums, and throat inflammations. So their tissue tightening the secretion reducing effects make them useful in anti-diarrheal and anti-hemorrhoidal products, while their antioxidant action helps protect cells and promote faster healing in topical applications.
The experiment confirms that the sample contains tannins, as shown by its deep brown coloration, thick syrupy semi-dry consistency, and a semi-solid physical state. Additionally, it's soluble in water and alcohol but insoluble in either. These findings verify the chemical nature of tannins and their biological and pharmaceutical